Good afternoon, my dear friends. Today's our eminent resource person, Dr.A.SHAILAJA Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous, SIDDIPET. Madam has huge experience in the field of plant sciences. Madam has explained in previous class about the Anthoceras and Markantia type studies belongs to BRYOPHYTA. So these are the most important topics for competitive exams. Along with that, today Madam is going to explain about the BRYOPHYTA type study, that is POLYTRICHUM. So it is also most important topic, especially the structure of the capsule, structure of the sporophyte, life cycle is most important. So listen carefully, my dear friends. Regarding of this class, if you have any doubt, interact with our respected madam. Madam, please welcome and start your session, madam. Please welcome.
Okay, sir. Thank you. Good afternoon, my dear students.
Madam. Hello.
Hello, sir.
Madam, proceed.
Okay. Okay, today we are going to start the BRYOPHYTA. So first, this is the third class of BRYOPHYTA. Advanced group of bryophytes are called masses. So Markantia are called liverworts and Anthoceras are called hornworts. And these BRYOPHYTA members are called masses. These masses are included in the class BRYOPHYTA. Gametophyte is differentiated into rhizoid, stem, and leaf. Screen is visible, sir? So, these BRYOPHYTA gametophyte is differentiated into rhizoid, stem, and leaf. First, I will explain that what are the important points of a BRYOPHYTA member. So, this plant shows two substages in the gametophytic phases, that is protonema and gametophore stage. See here, I will show you in the diagram. See here, this is the male and female. Okay, no problem. So in this plant, we are observed that two stages in the masses, two substages are there in the gametophytic phase, protonema stage and gametophore stage. The first phase of the gametophyte is filamentous. It is called protonema. Okay, which is temporary stage, that is erect branches arising from the protonema are called gametophores. With the development of these branches, the protonema gradually disintegrates. The gametophores develop into independent plants which are differentiated into rhizoids, stems, and leaves. The leaf-like structures are called phylloid protonema, resembling filamentous algal forms. This is the protonema. Okay, the first phase of the gametophyte is filamentous, that is called protonema. After that, that is the temporary stage. So when the erect branches arising from the protonema, these are called gametophores. Two substages are there in the gametophytic phase. These stages we are observed in the only BRYOPHYTA members. This is the developed members in the bryophytes. So that's why we are observed the two substages of protonema and gametophore in the gametophytic phase. Okay, all are you understood? That is protonema and gametophore. So the sporophyte of BRYOPHYTA members is distinguished into foot, seta, and capsule. In the sporophyte, same like that foot, seta, capsule are observed. In the Markantia, same Markantia sporophyte also having these three parts, foot, seta, capsule. But whenever the Anthoceratopsida, so that Anthoceras members having that sporophyte is distinguished into food and meristematic region or zone, and capsule is there. Theta is absent in the Anthoceratopsida. Okay, the capsule shows stomata also in the Bryophyta. So archisporium derived from outer walls of endothecium or inner walls of amphithecium. Capsule is differentiated into apophysis, theca, and operculum. This is the developmental stage in the sporophyte capsule. Okay, so I will discuss these deeply in sporophyte structure. The photosynthetic part of the capsule is called apophysis. The fetal part is called theca, and the apical part is called operculum or lid. Okay, this is about the Bryophyta general characters. These plants exhibit the heteromorphic alternation of generation type of life cycle. That is, these are all are belongs to Bryophyta members. Okay, so we are start the Polytrichum type study. So, what is the systematic position of Polytrichum? That is, see here, division Bryophyta, class Bryopsida, and subclass Bryidae, order Polytrichales, family Polytrichaceae, genus Polytrichum. See here, division Bryophyta, class Bryopsida, order Polytrichales, family Polytrichaceae, genus Polytrichum. Okay, this is the Polytrichum classification. So come to the gametophyte first occurrence. See here, Polytrichum have worldwide distribution. So in that, with totally 111 species are there in the Polytrichum. In our India, only 5 species are found, and common species are Polytrichum alpinum. They are very common in cool temperature and tropical regions. Plants live in cool and shady places. They may be found growing on open soil or tree trunks as epiphytes. This is about the occurrence of Polytrichum. Okay, totally 111 species are there. In our India, only 5 species are found. Polytrichum alpinum, this is a very common species. Okay. So next, coming to the gametophytic phase, see here. The main plant body is gametophyte. Okay, gametophyte. The adult plant consists of 2 parts, rhizome and upright leafy shoot. First one is the rhizome. It is horizontal portion and grows underground. It bears 3 rows of small brown or colorless leaves. It also bears rhizoids. The cells are rich in protoplasm and oil globules. That is about the rhizome. Okay. So in the gametophytic plant, that 2 substages are there. First is the protonema stage, and the second is the gametophore stage. In the favorable conditions, when the spore germinates and develops into a filamentous structure, that is which is grows horizontally. This is a branched septate form called protonema stage. Okay, so I will show you what is the protonema part and what is the gametophore. After the sporophyte germination, we will observe that. Okay, see here. This is the life cycle of Polytrichum. See here, this is the protonema. After this spore germination, whenever we are observed that that is the protonema stage that develops into a filamentous structure, grows horizontally. Okay, so this filamentous structure grows horizontally. That is called protonema. And which branches, like erect branches, are called gametophore. 2 substages are present in the Polytrichum. Are you understood? Jayant Reddy? Okay. Yes. So protonema stage and the gametophore stage. We are observed the 2 substages in the gametophytic phase. Okay. So that is upright leafy shoot. The leafy shoots are much longer. It is the most conspicuous parts of the plant. It arises from rhizome. These branches consist of central axis. These branches bear large leaves arranged spirally. Okay, leaves have broad bases. Leaves in upper portion are green, but the lower ones are brown color. Upper portion is green in color, but the lower part is brown in color. Each leaf has a broad, colorless sheathing leaf base and narrow distal limbs. The midrib forms the major part of the leaf. These leaves possess, you see here, this is a polytrichum plant, a male plant and a female plant. These are the rhizoids, these are the leaves. Okay. So we are discussing about this. We are coming to the leaf anatomy. See here, internal structure of leaf. So, okay, this is the leaf anatomy. Okay, this is the stem anatomy. This is the leaf anatomy. So you observe in the leaves show many celled thick midrib. See here, this is the midrib, and the lower epidermis of the leaf is continuous and distinct. The cells of the lower epidermis are large with a thick outer wall. This is a epidermis. The outer layer is called epidermis, lower epidermis, upper epidermis. So this is the single cell thickness, and next to the epidermis, 1-2 rows of sclerenchymatous tissue is present below the epidermis, and thin-walled parenchymatous tissue is spread in the midrib region. This is the parenchymatous region. This middle part of the leaf, and a few sclerenchymatous cell groups are also present in this parenchymatous tissue. Okay, sometimes we observe that some sclerenchymatous cells are present in the parenchymatous tissue. Rows of chlorophyllous filaments are present in the upper region. These are the chlorophyllous filaments. Okay, see here. Each row consists of 5 to 8 cells, and apical cell is always longer and bilobed. See here, this is the apical cell. So this is the longer cell than the below cells, and these large upper rows of cells form a discontinuous epidermis by arranging very close to one another. And chlorophyllous filaments are photosynthetic and also hold a thin film of water between them. The lateral part of the lower epidermis extend as wings. Okay, the lower part of the epidermis extend as wing-like structure. Okay, this is a wing. Okay, this is a leaf anatomy, and come to the stem anatomy, rhizome or stem anatomy. So same epidermis, cortex, and central cylinder. We are observed that epidermis, cortex, central cylinder. Okay, so in the rhizome structure and the same stem structure, stem anatomy and rhizome anatomy is the same. Epidermis, that is the one cell thick membrane without any intercellular spaces. See here, this is the epidermis. Outer layer is called epidermis, and inner to the epidermis, that is called cortex. The parenchymatous part below the epidermis is called cortex. 3 zones of thick walled cells are present in the cortex. These are called hypodermal strands. The ends of these strands gradually narrow into what are called radial strands. See here, this is a radial strands. Okay, in between that, below the epidermis, these radial strands are observed in the cortex region. Okay, so coming to the central cylinder, inner to the endodermis, in a 2 to 3 cell thick pericycle is there. These 3 radial strips are present. The inner structure of the pericycle is called central cylinder. Okay, this is the central cylinder. Leptome, hydrome, these are present in the stem anatomy. This is very important. What is the leptome? What is the? I will tell you. Okay, the outer layer of the cylinder, central cylinder, is called pericycle. Remember this. In the stem anatomy or rhizome anatomy, we are observed that is the pericycle. Okay, that outer layer of the central cylinder is called the pericycle. This basically shows the result, the food material, that is starch. That is the starch. Inner to the pericycle, sieve-like cells are present. Okay, see here, this is a pericycle. This single layer is called pericycle, and inner to that, these are called some sieve-like cells are present. These are called leptoid, and this group of cells is called leptome. Okay, some different cells are called sieve-like cells are present. That is called leptoid, and this group of cells is called leptome. These cells consist of starch, and hence the remaining cells are filled just with protoplast. They are termed leptome mantle. Okay. So inside the leptome mantle, starch-filled rows are present. That is, amylum cells are present. The center of the stem is occupied by dead cells. These are called hydroids and this group of cells is called hydrome. See here, leptome, hydrome. Hydrome is the center. Okay. So the center of the stem occupied by dead cells. These are called hydroids, and this group of cells is called hydrome. Okay. Inner to the pericycle, some sieve-like cells are present. These cells are called leptoid, and this group of cells leptome. These cells, leptome means it consists of starch, and hence these remaining cells are filled just with protoplasm. They are termed leptome mantle. Okay, remember that. What is the leptome? It's filled with starch. Hydrome. So that is hydrome cells are dead cells are present in the center of the stem. These are called hydroids, and this group of cells is called hydrome. This is equivalent to the xylem of angiosperm. So when we are observed that xylem and phloem for the conduction of water and some food material supply, but in the this is a developed mosses in the BRYOPHYTA members. So that's why that started that what is the leptome and hydrome are observed in the BRYOPHYTA members. Okay, so like that xylem in these cells, that hydroids in this stem, we are observed that hydrome mantle. Okay, so this is the stem anatomy or rhizome anatomy. See here. So stem anatomy. See here. The two zones, central zone and peripheral zone. The cortex consists of thick walled cells, number pair of cortex. This is the stem anatomy. Next, coming to the reproduction. So vegetative reproduction, it takes place by following methods. What are the protonema and vegetative buds and the fragmentation? Okay, the spores germinate to form protonema. Several buds grow on the protonema. Each bud by of its apical cell develops into gametophyte. These are the vegetative methods. Remember this. Mainly sexual reproduction is very, very important. Polytrichum is dioecious. Okay, Antheridia and Orchigonia occur in different plants. So that's why we are called dioecious. Next one is the antheradial and sexual reproduction. In the sexual reproduction, that male gametangia is called antheridium, female gametangia is called archegonium. Antheridia is club-shaped structure and the archegonium is flask-shaped structure. Okay, Polytrichum is a unisexual plant that is dioecious. The male and female reproductive structures develop on different gametophytic plants. Okay, remember that. That is the important thing. See here, I will go to antheridium. This is the antheridia, club-shaped structures. So this is the male reproductive organs. In the polytrichum, the antheridia are produced in clusters at the apices of the main branches. Special leaves are called perigonial leaves. See here, these are called perigonial leaves. Okay, so in this pericycle and perigonial leaves are present in the polytrichum. Okay, in the BRYOPHYTA members, we are observed that that is the protective layers. Okay, in the polytrichum, the antheridia are produced in clusters at the apices of the main branches. That special leaves are called perigonial leaves. Encircle the antheridia and give a flower-like appearance. See here. These perigonial leaves are arranged like this. This is a look like a flower-like appearance we are observed in the polytrichum. Okay. So these due to this, it is referred as antheridial flower. The longitudinal section of the antheridial branch apex shows paraphysis between the antheridia. See here. These are the paraphysis. These are the club-shaped antheridias. Fully matured antheridia are club-shaped with multicellular stalks. Antheridium is divided into many chambers. Antheridium is divided into many chambers. See here, this is the antheridium structure. Each antheridium is developed from a single cell. This cell is called antheridial initial. Okay. So this is the stalk. This is the antheridium club-shaped structure. Many cells are present in this. The maturity of the antheridium, that perigonial leaves are observed. The antheridial wall and its cells, due to the presence of extracted, the lead cells ruptured and release the male gametes. Okay, so that Lid cells are ruptured from this perigonial leaves and they release the male gametes. That is about the antheridia. Okay. So next coming to the this is archegonia. See here, this is a flask-shaped structure. Archegonia develop in group at the apices of the special branches in polytrichum. These branches are called archegonial branches. In each group, 3 to 6 archegonia are present. See here, 3 to 6 archegonia. Okay, these are the paraphysis, and this is the archegonium. Okay, in between the archegonium, that paraphysis are present. Sometimes we observed in each group 3 to 6 archegonia are present. Pericytial leaves are present around the archegonia. These are called pericytial or pericytial leaves. Okay, so remember these are very important. In the polytrichum antheridia, we are observed that surround the antheridium, perigonial leaves are present. In the archegonia, pericytial leaves are present. Okay, so this entire structure resembles a flower because of which it is called the mass male flower, female flower. Sorry, this is a mass female flower. Each archegonium has a flash-shaped structure differentiated into two distinct parts, the neck and the venter. See here, this is the neck, this is the venter. The neck is an elongated part with 6 neck canal cells and 4 lid cells. See here, these are the 6 neck canal cells. So in the anthoceros also 4 to 6 neck canal cells. It's very important. In the Markantia only 4 neck canal cells, but in the polytrichum 6 neck canal cells and 4 lid cells. Lid cells are common. The venter is of a broad globose form with 1 venter canal cell and 1 egg cell. See here, this is the venter canal cell. This is the venter canal cell. This is the egg cell. These are the neck canal cells. Okay. So each archegonium develops from a single cell, that is archegonial initial. After the development of archegonia, the primary venter canal cell divides to develop into the venter canal cell and 1 egg cell. These all are these neck canal cells and venter canal cells all are dissolved and form the one of the chemical substance formed in that neck canal. So that egg cell is present when that male gametes are entered into the archegonium and fertilized with the egg cell. So as the archegonia develops completely, the neck canal cells and the venter canal cells all are disintegrated. Okay, all are disintegrated. The lid cells are separated, and one of the mucilaginous chemical substance is released from the cells, and this mucilaginous substance attracts the anthrozoids. When that anthrozoid or male gametes are entered into the archegonium and fuses with the egg cell and fertilizes into it and forms the zygote. This type of fertilization is called chemotactic type of fertilization. Okay, this is a chemotactic fertilization. So this is about the archegonia and fertilization. Okay. So the sex organ stays in the presence of water. The venter canal cell and neck canal cells dissolve to form the mucilage or chemical substance. Okay, form the chemical substance. This mucilage exerts the pressure and the neck opens out. The mucilage comes out of the neck. The substance reached the archegonial heads by rainwater. So this is the fertilization. Next, coming to the sporophyte structure. Okay, sporophyte. See here. This is the sporophyte. It's very important. Sporophyte structure. So after the fertilization of male and female gametes in the venter of archegonium, the resulting zygote grows and develops into the sporophyte. This is in diploid condition. So diploid state, it comes of food, theta, and capsule. Okay, food, theta, and capsule. The food is embedded in the archegonia for branch tissue, absorbs the nutrients and water from the gametophyte and supplies them to the capsule through the theta. The food is parenchymatous and swollen. This is the theta is there. Food is below the theta. Okay. So that is the foot. It is Rani madam చేస్తారు ఏంటిది. Okay, so this foot is embedded in the archegonial branch tissue. It absorbs the nutrients and water from the gametophyte and supplies them to the capsule through the theta. That is the work of the function of the foot. The foot is parenchymatous and swollen. That is the first part. Next is the theta. This is a linear, long, and soft, and we have a capsule at the apex. In transverse section, it resembles stem anatomy. The outer rows of cells possess thick walls and another layer of sclerenchymatous strips. It gives mechanical strength to the theta. Nutrients are absorbed by the foot or transported to the capsule through the conducting cells of the theta. So, see here, this is the theta. This middle are the conducting cells. So when that foot absorbs the nutrients and minerals from the gametophyte, those are transferred from the theta to the capsule. Okay, that is the function of theta. And it gives the mechanical strength to the capsule. Next is the capsule. So this totally this structure is called capsule. At a young stage, the capsule is enclosed by the calyptra. See here, this is the calyptra. Okay, this is one layer that is called calyptra, and which is derived from the archegonial wall. As the capsule matures, the theta elongates and ruptures the calyptra layer. The calyptra remains as a hood or cap-like structure on the tip of the capsule. Okay, so this is the epithelium. Sorry, above the epithelium, this hood-like structure. Okay, that is the main calyptra. Okay, so we are called that is calyptra. When the capsule matures, the theta elongates and ruptures the calyptra layer. So then it is that remains structure as a hood or cap-like structure present at the capsule. Okay, around the capsule. So this at tip of the capsule, that hood-like structure is coming to the capsule. Once the capsule fully matures, this cap will fall off. The capsule is quadrangular in shape and it consists of three important parts. That is, in the capsule, three parts are there: apophysis, theca, and operculum. So remember this. In the Markantia and Anthoceras, these parts are not there to the sporophyte. But in the Polytrichum, in the capsule, three parts are there: apophysis, theca, and operculum. Apophysis, the basal part of the capsule attached to the theta, is called apophysis. So this see here, this is the apophysis, and the fertile part is called as theca, and this is the cap-like structure which we are called operculum. Okay, theca, apophysis, operculum. Apophysis, theca, operculum. These three parts we are observed in the capsule. That is the apophysis. The basal part of the capsule attached to the theta is called apophysis. A construction is present but between the apophysis and the theca part of the capsule. Theca part and the theta, in between this apophysis. Okay, distinct epidermis and stomata are seen in the apophysis region. Next to the epidermis, sclerenchymatous tissue is present. Apophysis of polytrichum is not as extensive as in Funaria. Okay, Funaria also belongs to this BRYOPHYTA member, and we are observed that this apophysis of polytrichum is not as extensive as in Funaria. Okay, this is the apophysis. Next one is the theca part, the middle fertile region. This fertile region is called theca, and the wall of the theca consists of several layers of chlorophyll cells. See here, these are the chlorophyll cells. Left side and right side, we are observed that the outermost wall forms the epidermis, and air chambers or air space, or also we are observed that lacuna is present inner to the wall layer. See here, this is the air lacuna air spaces. Okay, between this, this is the photosynthetic layer. It is divided into small chambers by transverse filaments of chlorophyll containing the cells, that trabeculae. we are called trabeculae. See here, trabeculae. Okay, so diagram, please clearly observe that see here. This is the trabeculae. Okay, so which one that lacuna is present inner to the wall layers? These are the lacuna spaces, and these are it is divided into small chambers by transverse sections or transverse filaments of chlorophyll containing cells. That is, they are called trabeculae. And one end of the trabeculae is connected with the inner wall layer, and the other with the outer wall of the spore sac. Okay, one is the connected with spore sac, and one is the connected with outer layer. Okay, that is the trabeculae. The central part of the capsule is occupied by a thick column of parenchyma cells. This is a parenchyma cells. It is called columella. Okay, this middle center of the capsule. This is a thick column of parenchyma cells. That columella. This is a sterile tissue which is continuous with the central axis of the apophysis. Spore sacs are enriched by 2 rows of thin walled cells. The spore sac consists of 4 to 6 rows as sporial tissue. These cells undergo reduction division and produce the haploid spores. See here, these are the spore sacs. Okay, so left side and right side, outer air space and inner air space. This is in between that capsule, the columella is there. This is the capsule wall. This is the spore sac, outer wall and inner wall. This is the stomata. This is the apophysis. Okay, see here. So then the spore sac consists of 4 to 6 rows of cells or the sporal tissue. Then these cells undergo reduction division and forms the haploid spores. That is the capsule. And the next one is the operculum. So on the capsule, a cone-like lid is present. See here, this is the cone-like lid. Okay, this is we are called operculum. Okay, ఏదైతే క్యాప్ లాగా ఉందో స్ట్రక్చర్ that is called operculum. The margin of the lid comprises of 3 to 4 layers of thick walled cells in the form of a diaphragm. See here, this is the epigram, and in the side is we are observed that it is peristome. Okay, so these annulus is not distinct. Internally, the lower part of the lid has a transverse membranous tissue. It encloses the capsule and is called epiphragm. To the epiphragm, 30 to 64 triangle shaped peristomial teeth are present. See here, this is the epiphragm. So these 30 to 64 triangle shaped peristomial teeth are present. That is important. Please remember this. In the Markantia, elatus is the one special character. In the Anthoceras, pseudo-elatus are there. But in the Polytrichum capsule, that 3 main parts are there in the capsule: apophysis, theca, and operculum. So in the operculum, that peristomial teeth is there. 30 to 64 triangle shaped peristomial teeth are present in the operculum to the epiphragm. Okay, the lower part of these peristomial teeth is attached to the middle of the capsule, while the terminal part is attached to the margin of epiphragm. When the thin walled cells between the teeth dry up, a wall of pores are developed at the epiphragm. So the columella in the middle of the capsule and the operculum form a broad wheel-like structure. This epiphragm is present on this structure. Unlike cinerea, the peristomial teeth of Polytrichum are not hygroscopic and do not show hygroscopic movement. That is peristomial teeth. Okay, they will not showing the hygroscopic movement. So because of that, they are not hygroscopic in nature. So that's why they do not show hygroscopic movement. That is the peristomial teeth in the operculum. These to the epiphragm, these are arranged triangle shaped peristomial teeth. So next one is the spore dispersal. At maturity, the lid separates from the capsule. This is the lid. Okay. So when that at maturity time, that due to its hygroscopic in nature, the theta exhibits during movement, that the capsule moves back and forth. As a result of these heavy movements, the pores developed between the peristomial teeth are released. This method of spore dispersal is called sensor mechanism. So whenever the lid is opening, that peristomial teeth, these are coming outside. Okay. So this is the pores are developed between the peristomial teeth. These are released. This type of method of spore dispersal is called a sensor mechanism. That is very important. What is the spore dispersal mechanism in the polytrichum? We are observed that is the sensor mechanism. Okay, the capsule moves back and forth. Okay, these moves, that is a sensor mechanism. They are called a sensor mechanism. So germination of spore, when the spore falls on the suitable substratum, it germinates into a branched filamentous structure. It is called protonema. The upright branches of the protonema are green and develop into the gametophore. Tell me.
Ma'am, the class is up to 4:30, ma'am.
No, ma. Just in 2 minutes, I will close this session.
Okay, ma'am. My health is not nice, ma'am. That's why I'm
Okay,
asking.
okay. No problem, no problem. Just 2 minutes, I will close this. This is about the sporophyte. Then the upright branches of the protonema are green and develop into the gametophore. The underground branches of the protonema are colorless and form the rhizoids. Okay, so that gametophore develops as a gametophyte. This is about the polytrichum sporophyte. Are you understood? Any doubts?
Yes, ma'am, I understood, ma'am.
Okay, next, the last that what is the alternation of generation? So that is the life cycle of polytrichum. So polytrichum shows
Ma'am,
the
about sporophyte, you'll ask, ma'am, later questions.
yeah, the type study that the sporophyte is lost gametophytic structure and the sporophyte. After this
Compulsory they'll give one question or two questions, ma'am.
Bryophyte నుంచి 1 or 2 questions.
Okay, ma'am.
See here. In this
Ma'am,
polytrichum,
ma'am, your explanation is nice, ma'am.
thank you. If you have any doubt,
No, ma'am. At last, I'll ask, ma'am.
okay. Polytrichum shows heteromorphic alternation of generation. Same in the Markantia and Anthoceras also. This is the gametophyte and the sporophyte. So see here, the plant body gametophyte, then in the gametophyte that develops the antheridia and archegonia. So then after that fertilization and going to form the zygote. After the zygote development, sporophyte is developed from the zygote. So next, after the sporophyte development, the spores, haploid spores are released from the sporophyte. These spores germinate and produce the protonema stage. Then gametophytic stage is developed from that. Okay, that is the alternation of generation. ఇక్కడ చూడండి ఈ విధంగా మనకి so the life cycle of polytrichum is called heteromorphic alternation of generation because that haploid gametophytic stage is there and diploid sporophytic generation is there. So that's why we are called heteromorphic. Different types gametophyte sporophyte కాబట్టి so heteromorphic alternation of generation 1 by 1 వస్తుంది కనుక we are called heteromorphic alternation of generation. This is about the polytrichum. Okay, completed. Thank you.
Okay, ma'am.
So now you will ask some if you have any doubts, please ask me now.
Okay, ma'am. Ma'am, firstly you told no, ma'am. Systematic position,
Okay, my dear
division,
friends,
order.
regarding of this
Yes.
class,
Naina,
if you
tell me.
have any doubt, please ask our respected madam.
Yes, sir. I'm asking, sir.
Okay. Systematic position of polytrichum.
Yes, ma'am. In systematic position, you said that division, class, order, family, and genus. In the 5 families, first one is Bryophyta. Am I right, ma'am?
Division Bryophyta.
Yes, ma'am. And second one is Bryopsida.
Class.
And third one is polytrichales and family is Polytrichaceae and genus is Polytrichum.
Yes.
Ma'am, what is the meaning of polytrichales, ma'am?
Polytrichales, it is one of the order.
Order, ma'am.
So we are dividing the plants as the systematic position. We are giving to the any plant that we are giving the one of the systematic position. Okay. So that is the Polytrichum systematic position in the plant kingdom. Okay.
Okay, ma'am.
The Polytrichum plant belongs to division Bryophyta, class Bryopsida, order Polytrichales, family Polytrichaceae, genus Polytrichum. Okay.
Okay, ma'am. And these two diagrams and reproduction, these all I understood, ma'am.
Okay.
And
So mainly, what are the special features about the Polytrichum? That the perigonial, perichetial leaves are present around the antheridia and archegonia. And in the sporophyte structure is very important in the Polytrichum. Because of that, food seta capsule, that capsule has the three parts, that is apophysis, theca, and operculum. Okay. So apophysis, theca, and operculum. Theca is the fertile part in the Polytrichum capsule.
Okay.
Okay. Remember that. Note down these all points. It's very important. The peristomial teeth are present in the operculum. 30 to 64 peristomial teeth is present in the operculum.
Ma'am, you explained already this sporophyte one class, no, ma'am.
Just in the general characters we are discussing them.
Ma'am, now you explain what, ma'am?
Polytrichum type study today.
Okay, ma'am.
So 3 type studies we learned. Okay. One belongs to hepatic apsida, one belongs to anthoceratopsida, one belongs to bryopsida.
Okay, ma'am.
Okay.
Okay, ma'am.
Okay, sir. Thank you.
Okay, madam, thank you very much. So, my dear friends, each type study consists two distinct phases, that is gametophytic phase and sporophytic phase. Okay. So, madam has clearly explained each type study, including Markantia, hepatic, anthoceras, and today's topic, that is polytrichum. So, these type studies consist special distinct phases. Two phases are here. One is gametophytic phase and the second one is sporophytic phase. Each stage is alternately comes. So that's why madam has clearly explained about the alternations of generation, the life cycle of the entire bryophyte studies. Madam has clearly explained. And most important in bryophytes, the structure of the archegonium consists of lid cells and ventral canal cells and ventral cells. These are the most important. Number of cells are most important. Along with the sporophyte, neck canal cells. Along with the sporophytic structure, especially sporophytic structure, what is the difference between the foot, seat, and capsule? So these are the most important. So thank you very much, madam. Tomorrow we will meet in same time along with gametophytic evolution and sporophytic evolution.
Yes, sir.
Thank you.